Hello, everyone. Welcome back. I thought I'd start with what we used to do when we were physically together. Take a look at the current situation, which has really amazingly changed. We have over 700,000 global cases. Now the U.S., of course, is number one in global cases. I think this is about a tenfold underestimate because we're not testing very much for the daily situation report. And this gives you a nice epidemic curve of all the WHO regions. And so we started the course around here. Look at that. It was hardly anything. And this is the China experience. And this is the rest of the world. I don't know when this is going to end, but it's not going to be forever. I think it will be probably 2 months maximum, but we'll see. Today we're going to talk about acute infections. We actually have two different kinds of infections we're going to talk about in the next two lectures this week, acute and persistent. And this graph summarizes both of them. So here we have virus production on the y-axis and time on the x-axis. And the blue line is virus production, and then the red blocks are periods of disease. And so on the top is our typical acute infection. That's what we'll talk about today. Rhinoviruses, rotaviruses, influenza viruses, poliovirus, SARS coronaviruses. A defined period of virus production, disease, and then resolution. And that's it. In contrast, there are many viruses that cause persistent infections, and these are different types of persistent infections we'll talk about more next time, but basically they can last the lifetime of the host. There can be periods of disease. There can be no disease and persistent production of virus. All of us have JC virus in us. It's replicating all the time. No disease. And this is HIV on the bottom. Initial disease, a long period of virus production at low levels, and then death. But today we'll do acute infections. And these are characterized by rapid onset of viral reproduction, short but often severe course of disease, the making of a lot of virus particles, and eventually immune clearance. And here's a typical time course in a little more detail mixed with what the host is doing. Again, virus reproduction on the y-axis and time on the x. And here we have the beginning of the infection. At some point, the virus begins to replicate. That's shown in the red curve here. Early on, we have innate defenses arising. We talked about those last week. And then those innate defenses may clear the infection. In fact, most virus infections are cleared by innate defenses. We don't even know we have them. But sometimes the virus overcomes innate defenses, maybe by having an antagonist or multiple antagonists. Virus continues to replicate. And then at some point, we activate the adaptive response. And of course, then there's a bit of a delay, 1 or 2 weeks before antibodies and T cells are made. And the replication of the virus peaks, the adaptive responses kick in. And in particular, CTLs may help clear the infection. Virus titers go down. And then we are left with immune memory, which we talked a little bit about last time. And the virus is typically cleared, although now we recognize that some acute infections may go on a bit longer. This little dotted line represents that. Eventually, they're going to be down to zero. But because we have sensitive methods like PCR, we now recognize that viral genomes persist a long time past what we used to think they did. And that's the case with SARS-CoV-2. We're seeing patients who are released and they apparently are PCR positive, but we don't know if it's infectious virus. I suspect it's not after a certain point, and it's just pieces of nucleic acids that are remaining. A key point about acute infections is that many of them are inapparent. These are successful infections. The virus is replicating, it's spreading, but there's no symptoms and no disease. Asymptomatic infections. And the key here is that the infected host makes enough virus particles so that the virus will spread in the population. So this is a problem with the current SARS-CoV-2. We think there are a lot of asymptomatic or mildly symptomatic infections that are spreading the infection. How do we know that there's a such thing as an asymptomatic infection? Well, we do serological surveys, mostly. In this very large outbreak, a number of people have been tested who had no symptoms, and they turned up to be positive. We're not doing that enough in the U.S. because we don't have enough test kits. But if we could test more people, we would see a lot of these asymptomatic infections. Typically, after the outbreak is over, we do serological surveys. We take serum from people and we measure it for antibodies against the virus. And these serological assays are just coming online in the U.S., and soon we're going to start to know how many people are infected. Very extensive studies will be done to know what fraction of the population is infected, and that's why I think that number of infections will go up tenfold when we're done. These viruses that cause asymptomatic infections are what I consider well-adapted pathogens. For example, over 90% of poliovirus infections are inapparent or asymptomatic. A very small fraction cause disease. In my view, that's a well-adapted pathogen because it spreads efficiently from infected person to person and causes very little disease. In my view, there's no need to make disease. It's basically a response of the host in many cases. But what's needed is for the virus to spread from host to host. And so I have here SARS-CoV-2 question because the extent of inapparent infections remains to be determined after this outbreak is over. Acute infections are typically common public health problems. They cause outbreaks affecting millions of people each year. Influenza viruses, noroviruses cause millions of infections. Most people don't hear about them. And you'll see this year the burden of influenza is huge. SARS-CoV-2 is getting attention because it's spreading globally. It's very visible. There are going to be over 1 million infections, probably tens of millions of infections when we're done. And these are problems because by the time you feel sick, the infection may be over and it's already spread to someone else. They're so rapid. And the asymptomatic character is a problem. By the time you feel sick, the infection has probably spread to someone else. Okay, first question. Which of the following do acute infections and incubation periods have in common? Virus is not replicating. No symptoms are visible. Immune defenses are engaged. The immune system does not respond, or all of the above. Okay, let's have a look. The answer is immune defenses are engaged, which I think it's the first time not everyone got it. A lot of you got no symptoms are visible, but a symptom is not visible. A symptom is only what you can feel. So that's kind of a throwaway there. It is the virus is replicating, the immune system is responding, and immune defenses are definitely engaged. What I want to do now is to talk about a set of viruses that cause acute infection and go through their properties and how they infect and transmit and so forth. Learn something about the generalities of acute infections. And we're going to look at influenza virus and measles virus as examples of viruses, respiratory transmitted viruses. We're going to look at poliovirus and norovirus as examples of viruses transmitted by fecal oral routes. And then we'll look at a mosquito transmitted virus, West Nile virus. And I thought about including a coronavirus here, but we don't have enough information on the current one. There are 3 types of influenza viruses. We call them A, B, and C. Remember, these are enveloped viruses with a segmented negative strand RNA genome, glycoproteins in the membrane, hemagglutinin and neuraminidase. Here's a CDC view, 3-dimensional reconstruction with it cut open so you can see the ribonucleoproteins inside. Types A, B, and C. Now, type A and B cause a very similar respiratory disease. Type C, mostly inapparent or mild upper tract illness. We vaccinate only against type A and B, and only type A causes pandemics. A pandemic is what we're having now for SARS-CoV-2. And every 20 or 30 years, a new influenza A virus arises that causes a pandemic, very much like what's going on now for SARS-CoV-2. The last one was 2009, and we'll talk about that later when we talk about emergence. The hallmark of these viruses is antigenic variation. They change as frequently as year to year, necessitating us to change the vaccine. These viruses are transmitted by droplets, respiratory droplets produced by coughing, sneezing, and talking, typically these large and smaller ones that don't go very far. It's also transmitted by direct contact with infected individuals or contact with contaminated surfaces. You then touch your mouth and eyes and nose, very much like SARS-CoV-2. The virus enters the respiratory tract. It replicates in the mucosal surfaces, the epithelial cells, and spreads all the way down into the lung. Of course, once it's replicating in the epithelial cells, it can then be exhaled and transmitted to others. So here's a blow-up of the tract, the epithelial layer, the basement membrane. The virus is well contained within the epithelium, does not establish a viremia, but it can infect the upper, middle, and lower tract. It can go throughout the tract. Sometimes you can get a mild upper tract influenza. Typically, it's focused in the trachea. You often have substernal pain with influenza, and in some cases can go all the way down into the alveoli and cause influenza pneumonia. Here's a closer look at the respiratory tract with an emphasis on the receptors, the sialic acids that are the receptors for influenza virus, and where they are distributed. And remember, alpha-2,6 linked sialic acids are receptors for human influenza viruses, while alpha-2,3 linked sialic acids are receptors for avian influenza viruses. So avian influenza viruses, as the name says, infect birds, and there are high pathogenic variants that cause extensive destruction of bird populations, and occasionally they can get into people, especially people who care for chickens. And so we have receptors for those viruses. They've never established extensive human-to-human transmission, but we're afraid of such an event happening one day. And if you look at the respiratory tract, you can see on this picture the sialic acid receptors are indicated. So up in the eye area, there are alpha-2,3 receptors on the cornea and the conjunctiva. So human viruses would not likely infect there. But there have been occasionally reports of avian influenza outbreaks caused by infection here in the eye with alpha-2,3. There is a lacrimal duct that drains tears into the nose, the nasolacrimal duct, and that actually has alpha-2,6 and alpha-2,3 receptors. So in principle, you could get some human influenza virus in your eye, and it would get washed down into your nose and infect there. The nasal mucosa, the paranasal sinuses, these have alpha-2,6 receptors, a common place for influenza virus. Human influenza virus to infect the pharynx, the larynx, and then throughout the rest of the tract, there are alpha-2,6 receptors, and the alpha-2,3s are typically deeper in the lung. Trachea, the bronchi, and the alveoli have alpha-2,3. So if the avian influenza viruses get deep into the lung, they can also initiate an infection. Uncomplicated influenza, short incubation period, which depends on how much virus you acquire, is a big discussion point for SARS-CoV-2. Yes, the dose matters. If you get one droplet with virus in it, it's going to have a longer incubation period. It might not take at all because your innate system might take care of it. It's usually an abrupt onset. You can tell the day you got influenza, headache, chills, dry cough, followed by high fever, muscle aches, malaise, and anorexia. Fever peaks within 24 hours, quite high. It starts to decline. It's gone by day 6, but as it declines, your respiratory system's signs intensify. You have a cough that changes from dry to productive, meaning you're coughing up mucus, often from deep parts of the lung, and this cough and weakness can persist for 1 or 2 weeks. During the clearing of infection, cytotoxic T lymphocytes are lysing infected cells in the trachea and the bronchi, and that causes a destruction which can cause the cough and the pain, substernal pain. We diagnose influenza in a number of ways. In the winter, in temperate climates like ours, we call it influenza-like illness. Fever, cough, or sore throat. You go to a physician. If you have those symptoms, they will give you an antiviral without even testing you. There are rapid lab tests, dipstick-style tests, which can be done in a doctor's office. They only have about a 50 to 70% accuracy. In other words, if you're positive, if you're actually infected, half the time the test won't pick it up. So they need to be improved. Better assays are PCR, culturing virus, serology, but most physician offices, of course, don't do that. Those are done by labs, and they take longer. Here are a couple of graphs of experimental infections to give you an idea of how the infection proceeds. So we can actually infect people and do these studies with mild strains of influenza virus, or we use people who have been immunized. And so what we do is here on the left, we've inoculated someone, we pipette some virus into their nose, and then at different days post-infection, we measure viral titers and nasal wash, and then you get a symptom score. You ask them how they feel on several counts, and you take their temperature and so forth. So 0 is inoculation. 1 day, you can see symptoms are pretty low, but we already have a good amount of virus here. It already peaks within 2 days after infection and then begins to decline immediately. So that's a beautiful acute infection, really quick. There's only about a day of shedding before symptom onset. And then the symptoms peak about a day later, and they both start to decline. You can see 9 days later, it's over. In this particular case, it's an average, I guess, of a number of individuals. On the right is a similar assay, a different study where we're looking at antibody titers on the left here and interferon titers. These are both in nasopharyngeal wash and virus here. So again, infection day 0, we have a rapid peak of virus titer. The febrile illness is indicated by the pluses here, up to 6 days. And then the virus titer goes down. Initially, high titers of interferon, you can see, matching virus titers, that goes down as well. And then the antibodies rise later. So these have no role in clearing the infection, obviously. They rise way later, but they, of course, establish memory. What's clearing the infection is a combination of innate and cytotoxic T lymphocyte responses. Let's take a look at influenza in the U.S. Now, we have an extensive tracking system for monitoring influenza, not only in the U.S., but in virtually every country of the world, because it's a really big deal. And all these labs feed into each other, and they share data. And this is from the CDC, which looks throughout the U.S., of course. And I have 4 years here, just to show you the seasonality of influenza in the U.S. And you can see starting at about week 40, the fall, going through the end of the year, and then ending typically week 20 or so in the spring. And every year, pretty much the same seasonality. In between seasons, so in the summer, there's not a lot of circulation, but it's not 0, as you can see. So the line is simply all the nasopharyngeal samples that were positive, the percent positive for influenza every year. This is a number of isolates on the left and the percent positive on the right. They're up to 30% are positive here. So you can see there's some infection in the summer months. So it is seasonal, but you don't have outbreaks in the summer months. You have the persistence of virus, which probably starts up the outbreak again in the fall. So that's the seasonality. Here are the numbers for this year, 2019-2020 flu season. These are preliminary. The season's not over. October through March. Between 38 and 54 million illnesses due to influenza. Medical visits, hospitalizations, 24,000 to 62,000 deaths. That's a lot. And this does not overburden the medical system as SARS-CoV-2 is right now. I think it's spread out, and maybe there are fewer of them in the end. We'll see. But this is a lot of illness and a lot of deaths. And we have a vaccine which could prevent a lot of this, but half the country doesn't take it. It's really weird because now people are clamoring for a SARS-CoV-2 vaccine. And my guess is in a couple of years, half the country won't take it anymore. Here's some more data to have a look at. First, on the right, lower right, here's a map of the U.S. This is a great page in CDC where you can go through every week of the flu season and look at the influenza-like illness activity level in every state. And so this is the most recent map. Red is high and green are lower. So you can say most states are still having high influenza-like illness throughout the U.S. Just a few have minimal. At the top, left and top and bottom, these are influenza-positive tests for this season on the bottom, 2019-20, and then the previous season for comparison. And we have the number of positive specimens with the week So starting at the beginning of the flu season in the fall here, and it's stratified according to type and subtype. A, H1N1. We have a couple of different A subtypes circulating. H1N1, which is the 2009 pandemic strain, it's still circulating. And H3N2, which is the 1968 pandemic strain, it's still circulating. And then we have a couple of B lineages here. So you can see this. Last year, the predominant isolate was 2009 H1N1, but there was also H3N2 circulating. And here, this is the 12th week. So that's like January, February, March, end of March. That season was pretty much over. Now this year, this is as far as we got here, end of March also. Similar peak, similar curve. Again, H1N1 predominating. So that's the seasonality. And finally, in the upper right, this is pneumonia and influenza mortality. So this is how influenza deaths are calculated, because not every death certificate has influenza on it. They often have just pneumonia, so you have to make an extrapolation. And so these two black lines are predicted lines throughout the year of the maximum and minimum flu and pneumonia deaths. And then the red is the actual data, the percent of all deaths due to pneumonia and influenza. So you can see some years are mild. Some years the number exceed the epidemic threshold that's calculated. Here in this year, 2017-18 was a very severe year for influenza. And this year so far, not very severe. So we have a lot of data for influenza, as you can see. And finally, the percentage of visits for influenza-like illness reported in the illness surveillance network in the U.S. So this is for multiple years, different colors. The current year is 2019-20. That's red. So we're looking at percent visits for influenza-like illness. So someone goes to a hospital or clinic and has influenza-like illness, and then with the week. So you see this year we've had a couple of peaks of activity, but these are specific for influenza. And in other years, some years it's been lower. So the orange, 2015, pretty low influenza year. This is the 2009 pandemic year in gray. Started much earlier than normally. It started at the end of the summer. Very unusual. And then by the winter, it had petered out. You can have complications of influenza. You can get pneumonia. The virus goes down into the alveoli, destroys cells, and the fluid moves in. You get pneumonia. You can get secondary bacterial pneumonia. And this is after you've recovered from influenza. The bacteria move in and cause a pneumonia, start to multiply. Generalized muscle pain. Sometimes the heart gets involved. If you're having difficulty breathing, you're not supplying enough oxygen to the blood, the heart will try and pump faster, and that can cause problems, especially if you have cardiac issues. And then a syndrome called Reye's syndrome, which involves encephalopathy and liver damage. This is not a consequence of virus replicating in the brain or in the liver. It's rather the consequence of soluble mediators that have moved to those areas and are causing damage. We do have some pharmacologic interventions, a lot of non-pharmaceutical things you can buy, but we do have prescription antiviral drugs, Tamiflu, Relenza, amantadine, and the latest one is Xofluza. And we will talk about how these work. They have distinct mechanisms of action when we talk about antivirals. And of course, we have a number of vaccines, which we'll talk about later as well. Next question. Which of the following is characteristic of uncomplicated influenza? Transmission may occur via respiratory droplets. Incubation period 1 to 5 days. Fever peaks within 24 hours. Coughing and weakness can last for 2 weeks. All of the above. What's the difference between causing disease and causing a pandemic? Well, a pandemic is when many countries globally get infected. There can be disease. So maybe you're asking about epidemic and pandemic. An epidemic is an outbreak in a single country. But when you have epidemics in many countries, then it becomes a pandemic. What properties allow it to become widespread? Lack of population immunity. With influenza, you have a pandemic strain emerge. No one has immunity to it. It spreads globally. Same thing with SARS-CoV-2. How do they estimate the number of illnesses most of the surveillance is not just through medical providers. There are clinical labs as well. There are influenza network labs that all report to regional labs. It's a really extensive system. What causes flu to only subsist at small levels? By the end of the season, there's a buildup of population immunity, and there's temperature and humidity, which restricts transmission. All of that reduces it, but doesn't eliminate it. And then in the fall, the temperature drops, the humidity drops, and the transmission goes up. Density of people. That's another factor. People are inside most of the time. It's true, and that probably continues it. What's the difference between stomach flu and respiratory flu? Well, there's no such thing as stomach flu, but that's what it's called. Influenza is a virus that infects the respiratory tract. Stomach flu is a name for norovirus gastroenteritis. For some reason, it's gotten the name stomach flu, but it's not influenza. It's a different virus. Why does only A influenza cause pandemics? We think because it's the only one with an extensive non-human animal reservoir, birds and pigs and many other animals. So we'll talk about that in emergence. All right, what do we have here? How did we do? Everybody look at that. I give you some time and you all get it. Great. This is all correct. All right, next is measles virus. Measles, another respiratory virus. It's a member of the paramyxovirus family. Envelope negative strand RNA virus with a single RNA. Again, glycoproteins in the envelope, hemagglutinin, neuraminidase, fusion protein. This is one of the most contagious human viruses, the R0, about 15. In contrast, the SARS-CoV-2 is between 2 and 3. There is one serotype, and measles virus does not change. We're using the same measles vaccine that we used 50 years ago. Doesn't change. And you get infected, you get lifelong protection. The virus is transmitted by inhalation of respiratory droplets and aerosols. These viruses go in those very tiny aerosols that travel long distances. This can infect people many, many feet away. If we were in our lecture hall in Schermerhorn and I was in the front with measles, the people in the back would get infected. And here you have about 2 or 3 days before the rash, when you're maximally contagious, you're transmitting virus. You don't know you're sick until you get the rash. And in contrast to a lot of other acute infections, almost everyone who is infected shows signs of disease. Very few asymptomatic infections. The pathogenesis here is really interesting. So it's a respiratory virus. You inhale the droplets. They replicate in the upper and lower tract in the respiratory epithelium. This virus, unlike influenza, traverses the basement membrane. It causes enough inflammation so that the virus can get across, goes through the lymph into the blood, and establishes a viremia. And that blood brings the virus to multiple organs, including the brain. In some people, you get encephalitis, and of course the skin, where you get the characteristic rash. And here's the timeline for the infection. The incubation period, you get the primary viremia, secondary viremia spread to the skin, and then the prodromal period, where you're shedding virus. You get Koplik spots in the mouth. I'll show you those. You get fever, malaise. And remember, the prodrome, you have signs and symptoms, but they are not characteristic of the disease until you get to this period, the rash, and that's characteristic of measles. You're still shedding virus, but you're now making antibodies. About 3-week disease. The way this works is really interesting. So on the right here, we've focused in on the respiratory epithelium. So here the virus is coming into you. The virus particles are infecting the respiratory epithelium. We think the virus infects lymphocytes in the epithelium, not the actual epithelial cells. There are no receptors for this virus on the apical respiratory epithelium. So they infect immune cells, which are these orange and yellow things here. And then those immune cells move through the basement membrane with virus in them and establish the viremia. Okay, once you have a viremia, there are blood vessels underneath the basement membrane. The virus is brought there, And at the basolateral surface of the epithelial cells is a receptor for the virus, Nectin-4. Virus binds to it. It gets into those cells by the basolateral domain, replicates in them, and then are shed on the apical surface. So this is amazing because there are no apical receptors. So the initial infection doesn't result in shedding like it does for flu. You have to go through all the viremia, and the virus comes back to the basement membrane area. The virus gets into receptors on the basolateral domain, replicates and is shed. It's a really unusual means of transmission. So uncomplicated measles, high fever, runny nose, cough, conjunctivitis, Koplik spots during the prodrome. Here's a Koplik spots, these little white things in the mouth of this child. In the old days when we used to have a lot of measles, you could look in the mouth of a child who had a rash and look at these and say, this is measles. These are syncytia caused by virus replicating in the mucosal epithelium and causing the cells to fuse. And you get a rash from face to extremities, all over the bodies. Very typical rash. Sometimes you have complications. You can have encephalitis. 1 in 1,000 infected kids get encephalitis, bronchitis, pneumonia, ear infections. 1 to 2 in 1,000 kids will die of measles infection in the U.S. In countries with poor nutrition, it goes up to 28%, as we mentioned last time, because this impairs your immune system. A very long-term consequence is a disease called subacute sclerosing panencephalitis, SSPE. And about 1 in a million kids who recover from measles, 20, 30 years later, they get a progressive degenerative neurological disease. It's ultimately fatal. Apparently, some form of the virus is persisting in the brain. We don't know why. It's barely studied. It's so rare. But that's been eliminated by measles vaccination. And finally, this virus is very good at immunosuppression. As we talked about last time, replicating in immune cells, and that leads to secondary infections, the main cause of death in third world children who have poor nutrition. In the past year, it has been discovered that measles infection erases immune memory and what we call immune amnesia. This was a study done in the Netherlands. There's a community of individuals who do not vaccinate, and so their children get measles. Curiously, they're very happy to participate in medical studies. So they got 77 unvaccinated kids. They tracked them before and 2 months after measles infection. And then they collected serum from them, and they use a technique called VIR-scan. And what you do here is you take peptides representing thousands of pathogen epitopes. You put them on glass slides, and then you flow the serum over, and the antibodies will bind. Then you can track what's binding. And then you can display all of the epitopes. So these plots, these blobs are all the different, the thousands of epitopes before and after measles. And what they found is that measles infection causes elimination of between 11 and 73% of the antibody repertoire in these kids, which is caused by the virus infecting B and T memory cells and plasma cells, and it destroys them. You have no immune memory cells left. So look at this. These are the control kids before and after measles. Negative kids, no effect. Measles, mumps, rubella vaccine. You actually stimulate immune memory by having been protected against measles infection. And then the mild and severe kids, measles, you can see the antibodies that are binding the different pathogen epitopes go down. And that's just displayed in another way here with a dots, with a whisker plot. You can see the vaccinated kids are better than the baseline in the control population. And then you have a severe reduction in the kids who were not vaccinated and got measles. Another reason to get measles vaccine, it prevents this loss of immune memory. Now, they did find that in these kids, when they get reinfected, you can restore the immune memory. It's not destroyed forever, but still, why bother going through all that? So in the U.S., we used to have 3 to 4 million cases of measles a year, a lot of deaths, a lot of hospitalizations, and chronic disability from encephalitis. Virus infecting your brain is never good. In 2000, we stopped endemic transmission in the U.S. by the use of measles, mumps, rubella vaccine, which combines all three viruses. And one, however, in 1998, a bad report from the UK from Andrew Wakefield led to decreased immunization in many countries, which continues to this day. There were outbreaks in the UK and Ireland, and these viruses spread to the U.S. They're imported, and outbreaks occur in communities where immunization is dropping because the parents believe the Wakefield report, which has been discredited and withdrawn, but nevertheless, they don't immunize their kids. And, for example, 2018, big outbreak in Rockland County, New York. We introduced a measles vaccine in 1962-63, and it nearly eliminated measles. There's a persistent number of cases in people who do not vaccinate. And that's shown here. These are measles cases by year in the U.S. You can see they're different numbers depending on the year. And this past year, we had 1,282 cases in 31 states, 128 hospitalizations, 61 with pneumonia and encephalitis. That's the most number of cases since 1992. And most of them linked to recent outbreaks in New York, including that Rockland County. Most in people who were not vaccinated. 2018, there were 17 different outbreaks in New York, New Jersey, New York City, many in unvaccinated people in Orthodox Jewish communities. And the virus is introduced by travelers who brought the virus from Israel, where there was a large outbreak. 82 different people brought measles to the U.S. from other countries in 2018. This is a real problem. It's not just a U.S. problem. Globally, this is the distribution of measles by month in WHO region for these years. Just in 2019, over 400,000 cases globally, and big outbreak in 2018, 2019, mainly in African countries, the blue, that's the WHO African region, but in other countries as well. Many of these due to not vaccination because the countries can't afford it. And so this should be a goal of our country and many others to try and promote this vaccination. Many of these kids get very sick, they die, and have lifelong disabilities due to encephalitis. And in the past 6-month period, these are the countries that have seen the most measles. The darker the red, the more cases. You can see we have our share here in the U.S. There's some countries without any, and there's some countries with many, many cases. The top countries are Brazil, DRC, Philippines, Bangladesh, etc. Next question. Which of the following is a good reason to get measles vaccine? 1 in 1,000 chance of encephalitis, 1 in 1,000 chance of death. Each infected person spreads measles, and immunosuppression can lead to secondary infections. Okay, let's see. Can COVID be transmitted in the same way as measles aerosol? No, I think mostly it's not. There have been some cases where there's long-range transmission, but I think it's the minority. I think the major transmission is droplets and by contact. I think contact plays a bigger role than we know. Why does measles mutate so slowly? No, it doesn't proofread. That's a good point. All these RNA viruses make errors. Only coronaviruses have a proofreading protein. And, compare measles to flu. We think the proteins just can't handle the amino acid changes. How can the immune memory be restored? Well, when you get an influenza infection, you get common cold, you will just restore it because the cells were killed by measles, but they're not killing the stem cells. So you can always make lymphocytes. Yeah, all of the above is the right answer. These are all correct. Those are all good reasons to get measles vaccine to prevent encephalitis, measles, death, to prevent transmission, and to prevent immunosuppression. All right, so those are 2 respiratory pathogens. Now let's move to 2 fecally, orally transmitted viruses, and one is poliovirus, the agent of poliovirus. This is my favorite virus because I've worked most of my career on it. It is a small icosahedrally built particle with an RNA genome of plus polarity. And it initiates infection when you ingest fecally contaminated material. All of us have really poor hand hygiene, especially kids, and they're wont to have fecal contamination of their hands, and they will touch their mouths and swallow it. The virus passes through the stomach, goes into the intestines, and it's very hardy. It withstands the low pH of the stomach, the high pH of the small intestine, the bile salts, the proteases, completely unimpeded. Replicates in the intestinal mucosa, establishes a viremia, and in 99% of cases, that's the end of it. Virus is shed in the feces and transmits very effectively. In 1% of individuals, the virus gets into the central nervous system. It can do so through the muscle. Virus enters the motor end plate where there are receptors and enters the nerve and gets transported up to the motor neuron, or it could actually get across capillaries in the brain and spinal cord. And then it destroys enough neurons in the spinal cord and brain, the central nervous system, and you get paralysis. But that only happens in 1% of infections. Otherwise, this is a very exquisitely adapted pathogen. And you may ask, like, why is this? What's with these 1%? So you could do genome-wide association studies and look for SNPs in these people, and it's just being done. People are just starting to do that after all these years. So we'll know soon, I hope. A couple of lectures ago, I talked about a transgenic mouse model for polio that we made in our lab, and this is a example of the pathology the virus causes in the spinal cord. This is a mouse that had been paralyzed by injection, and this is a section of the spinal cord, and you can see these darkly staining cells. These are immune cells of some kind, infiltrating, trying to clear the infection. And then the green dots are poliovirus RNA replicating in neurons. These are very typical neuron morphologies, pyramidal shaped with axons and dendrites. You can see in this side view here, beautiful viral RNA and axons and dendrites. So that destroys the neuron. The virus replication probably destroys the neuron and causes the paralysis. This is one of my favorite figures from a 1950s textbook of medicine. It's a summary of data collected from many individuals with disease, and I like it because it's drawn by hand, which is what we used to do in those days. So here we have a number of different parameters measured. We have time here at the top and the bottom in days, and we have exposure to the virus and what happens. So we have temperature being monitored. You see there are 2 spikes of temperature. So there's this first 7-day incubation, then headache, sore throat, nausea. And in most people, that is actually the end of the infection. And then we're looking at virus in throat secretions. You see peaking of virus and feces. That goes on for a long time. Virus in the blood peaks here. And then virus in the CNS. So in a very small percentage of people, after that initial infection, the virus continues. It enters the CNS. So then you have a second spike in temperature, headache and nausea. And some people just have that and no paralysis, but in others you have paralysis. And correlating with the paralysis, you have virus in the central nervous system. And so that's the chronology of poliomyelitis. Humans are the only known reservoir of this virus, which makes it a candidate for eradication. And we're almost there, as we'll talk about in the vaccines lecture. Spread by fecal oral transmission. It peaks in warm months and temperate climates. We're not sure why. We think it has to do a lot to do with kids interacting more in the warm months. There is a long-term complication called post-polio, which will occur 30 to 40 years after the initial paralysis in about a quarter to 40% of the individuals. It's not an infectious process. There's no virus present. What we think happens is that you've lost a number of neurons as a consequence of polio. With therapy, sometimes you can regain the function of your limbs, but then as you age, you lose neurons naturally, and in people who have already lost so many, they can't compensate. So you have another bout of paralysis, essentially. We used to have a lot of polio in the U.S., and the peak was in the 50s in the post-war baby boomers. So this peak of paralysis in the kids born after World War II, just a lot of kids to infect. We introduced a vaccine in 1954, and then a second one in 1962, an inactivated and a replication-competent vaccine, and that eliminated polio in the U.S. The last case of paralysis, 1979. And we will talk about the progress in global eradication when we talk about vaccines. Now, here's a fun fact for you. In any 24-hour period, about 200 million people on Earth have gastroenteritis, which is vomiting and diarrhea, and the amount of diarrheal water that they pass equals the volume of water passing over Victoria Falls every minute. 65 million liters per minute of diarrhea. This was given to me by a gastroenterologist at Stanford. Who else would know this? And most of this is caused by norovirus, which is a small plus-stranded RNA virus. We haven't really talked about these very much. It's in the Calliciviridae family. It causes half of all foodborne outbreaks of gastroenteritis just in the U.S. alone. 48 million cases per year. All mostly norovirus, as you can see, some bacterial gastroenteritis and others like chemical toxins and parasites and so forth. So this is a big deal, and so a lot of people are studying it. It's even a bigger deal globally. And, you know, with the current SARS-CoV-2, people are asking how many virus particles do you need to get infected? We don't know that, but with norovirus, you can actually do experiments in people. You could feed them norovirus. So we've done this experiment. As few as 18 viral particles are enough to start norovirus gastroenteritis. So a head of a pin could infect 1,000 people. What another great fact. I call norovirus gastroenteritis a two-bucket disease because you're vomiting and you have diarrhea. This virus is spread fecal oral contamination like polio. They pass through the stomach like polio. They retain their infectivity. They infect the mucosal surfaces of the GI tract. They cause blunting of villi in the proximal jejunum just past the stomach. So here's an villi of uninfected person and infected. You can see they're blunted. We presume this has something to do with the diarrhea, but we don't know the basis for that, why you vomit, why you get diarrhea. The molecular mechanism isn't known. Up until a few years ago, we could not propagate noroviruses in the laboratory. It turns out you need bacteria to do that. And so we were taking diarrhea from patients and putting it through a 0.2 micron filter to get rid of the bacteria, right? To sterilize it. And then we'd throw it on cells in culture. It never grew. And then one day someone made a mistake and forgot to filter it, and voila, the virus was able to infect cells in culture as long as the bacteria are there. And somehow the bacteria facilitate uptake of the virus into cells. And here's an experiment where we're looking at replication versus days post-infection. And these are B cells in culture. It turns out that's a major site of replication of this virus. If you filter the virus, the stool filtrate is not infectious. If you leave it unfiltered, so it has bacteria in it, the virus will reproduce in B cells. And this is our current view of the pathogenesis of infection. Here's a diagram of the intestinal tract with our intestinal epithelial layer and the villi. The gut lumen is here in blue. And interspersed here we have M cells, those green ones. We also have very rare cells called tuft cells. And we think human noroviruses infect these intestinal epithelial cells. We think they infect macrophages in the subepithelial space, dendritic cells there, and T cells. And also, it's been shown that B cells can be infected as well. And so, of course, there are B cells beneath the epithelial sheet. They're there to make antibodies, of course, and apparently they can be infected by noroviruses. And so somehow the bacteria are facilitating infection of cells. Exactly how that happens isn't clear. Some clinical and epidemiological features of these infections, of course, these are transmitted by fecal-oral contamination, typically of food or food handling items. But it can also be spread by aerosol. When you vomit, you aerosolize the virus, and that can be swallowed. Contact with contaminated material, food, water, environmental contamination. Foods can be contaminated at the source, like oysters can pull in fecal contamination in the waters. Raspberries, if they're washed with contaminated water, or even in the fields and during preparation by food handlers who are shedding the virus. Short incubation period, sudden onset of vomiting and diarrhea, stomach pain. A pretty rapid duration of illness if you are healthy, immunocompromised a bit longer. And here, this is a key to the transmission. 30% of the infections are asymptomatic. You get this virus and you do not have gastroenteritis. So you could be a food handler and you're infected and shedding, and you go to work, you have no idea you're infected, you contaminate the food, and this is often how outbreaks occur. The virus shedding peaks a few days after illness, but it may persist for 56 days after you've gotten well. These people go back to work, of course. You can't just stay home for 56 days, right? Oh, wait, that's what we're doing. Immunity is short-term. You can get reinfected many times in your life, but each infection subsequently is milder. Humans are the reservoir of this virus, but we are suspecting that the viruses may infect non-human animals and they may become a new reservoir. We need to do more work to study that. The virus infects everyone, peaks in cold weather, although you can get outbreaks year-round. And these outbreaks occur in semi-closed environments like nursing homes, hospitals, cruise ships, military camps, schools, recreational activities, anything that favors person-to-person spread, the kind of stuff we can't be doing now as long as SARS-CoV-2 is spreading. Nothing to do about this infection. Make sure you don't get dehydrated. Most of the deaths occur in places where they don't properly hydrate the kids, and that's all there is to it. A vaccine is in development, which presumably will prevent many of these infections. Here's some data on where these infections occur. What settings? The majority restaurants and catered meals situations. Again, food handlers shedding virus. Retirement centers, nursing homes, even hospitals, schools, cruise ships. And the mode of transmission, mainly person to person, fecally contaminated material, 10% foodborne. Here's an article where an airplane had a sick person. They threw up and they aerosolized the virus and they passed it on to the crew. So they had to clean up the mess. And then those crew went on different flights and they infected other people as well. So the vomiting will aerosolize the virus, and you don't inhale it, but it comes into your mouth and you swallow it and start a new infection. You don't need many virus particles, 15 virus particles to do it. And finally, cruise ships, which one company calls the happiest place on earth. Here's just a number of outbreaks of norovirus gastroenteritis. Sometimes we don't know what it is. Might be something else. There are other viruses that can do this, by the way. But a lot of norovirus gastroenteritis on cruise ships. And now, of course, we see SARS-CoV-2 infections on cruise ships. I don't frankly know why you'd ever go on one. Why are they associated with cruise ships? It's only because we look for it. Health officials track illnesses on cruise ships, so we find outbreaks really quickly, close living quarters, and new passenger arrivals bring virus to other passengers. When there's an outbreak, they have to bring the ship back. Everyone gets off, and they have to sterilize the whole thing. And of course, sometimes the food handlers are shedding, although I think they're being more careful about that. What can you do to prevent it? Wash your hands. This is from the CDC. Rinse your fruits and vegetables well. Cook your shellfish thoroughly. If you eat raw oysters, which, it's not a bad thing to do because they're good, but taking a risk, so I don't do that anymore. Clean surfaces. Wash your laundry. If you're sick, don't prepare food. A lot of these are the same thing you want to do now to prevent SARS-CoV-2 infection. And don't eat the mints when you leave the restaurant, because nobody's going to restaurants these days, but when at some point we will go back, there are these mints by the cash register. Don't touch them, because studies have shown that they're contaminated with fecal bacteria. Let's see if there are any questions here before I go on. Here's where we ended. Why don't we have the pandemic strains of flu in all vaccines? Well, we don't know what they're going to be, so we're trying to make a universal flu vaccine that would catch anything that comes out. And I think that's possible, but it's still 5 to 10 years off. What about the measles proteins makes them so conserved? No idea. We don't know, because they're hemagglutinin, neuraminidase, and so does flu have an HANA. So we have no idea. May the second fever peak occur without any CNS pathology? Yes. You can have a very mild second fever, which is accompanied just by headache, and there's no CNS pathology. Are there any efforts to create pig avian influenza vaccines? Yes. So for pig farmers and chicken farmers, there are already influenza vaccines to prevent that. I don't know about bat Ebola vaccines. Instead, they're immunizing people with Ebola vaccines. But I can tell you, we'll actually talk about this later. There is a MERS coronavirus vaccine developed for camels in the Arabian Peninsula, which is how humans get the virus. And there's a Hendra virus vaccine for horses, because you get, in Australia, that's how the virus is transmitted to people. How is that norovirus infection ethical? They use medical students. Actually, they do, but it's ethical because everyone recovers. It doesn't cause any death. So they infect medical students on a Friday. You get diarrhea and vomiting Saturday and Sunday. By Monday, you're well. You can go back to class. You get $300. Is there a difference between the viral loads of symptomatic versus asymptomatic? There probably is. Don't know what it is. Sorry. Oh, raspberries, blueberries, strawberries. Yeah, it can be everything. I just had one only. If you touch the plastic of the mint, you can still get infected because, someone's going in there and pulling out a single one, and they're contaminating others. So the plastic can be contaminated as well. Okay, our last virus is one spread to people by mosquitoes. This is West Nile virus. It's a member of the flavivirus family, plus strand RNA viruses. Isolated in 1937 in Uganda. Has nothing to do with Egypt. Okay, Shanahan. Nothing to do with Egypt. It was not present in the Western Hemisphere until 1999 when it came first to New York. I think I told this story in an earlier lecture. The virus began spreading in the summer of 1999. Eventually, they sequenced its genome and found that it was identical to a virus from an Israeli goose. So somehow the virus got to the U.S. from Israel, probably in an airplane, possibly in a mosquito. This virus infects many birds, many mosquitoes, and vertebrates. The transmission cycle, well, first of all, the virus is enveloped plus stranded RNA genome with glycoproteins lying flat on the virus particle surface. This is a virus of birds, primarily, in nature. Mosquitoes spread the virus among birds. And in birds where the virus has been for many years, they're fine. But when the virus came to the U.S., a lot of birds were dying because they'd never seen the virus before. It had not evolved in them. And so that was one of the sentinels in New York City. People were finding dead crows on the streets. And in the Bronx Zoo, a lot of the birds were dying. And it was actually a pathologist in the Bronx Zoo who first figured out that something weird was going on. It's a great story. My colleague Dixon Despommier wrote a book called West Nile Story, which documents all of this. So this is a cycle in birds transmitted by mosquitoes, and occasionally the mosquitoes bite a human or a horse, and they are dead-end hosts. They get infected, they can get serious disease, but they don't spread it to anyone else. Even if a mosquito bit an infected human or horse, the viremia is not high enough for the mosquito to get enough virus to transmit it to another person. So these are dead-end or incidental hosts. In fact, in the U.S., think about this. There can be thousands of infections a year. Every one is a zoonotic crossover from a bird. It's a spillover from a bird to a human. It's a brand new infection. So Culex mosquitoes transmit this virus 3 to 14-day incubation. 20 to 30% of people develop a flu-like illness because fever, muscle aches, etc., which is called West Nile fever. 80% of the people have no symptoms. And again, we know that because we've done serological surveys. A lot of people have antibodies to the virus and they never were sick. 1 in 150 people develop neuroinvasive disease. The virus gets into the central nervous system. Last time we talked about how TNF-alpha is a culprit there and getting virus into the CNS, permeabilizing the blood-brain barrier. If you have neuroinvasive disease, you have headache, eye problems, muscle weakness, cognitive impairment. You can have polio-like flaccid paralysis, and this has a 10% mortality. And over half of these individuals have long-term neurological sequelae. They never recover from it. And so a vaccine for this is in the works. We actually have one for horses already because apparently racehorses are very expensive and you don't want to lose it to West Nile virus, so it's easier to make a vaccine for horses. There are actually a couple of vaccines for West Nile in horses. One of them is a DNA vaccine. So here's the pathogenesis. So a mosquito is biting a bird, and occasionally will deliver the virus to humans. Remember, the mosquitoes deliver the virus into the skin, to the area which is vascularized. The virus replicates in the Langerhans dendritic cells. These bring the virus to the lymph node, and there the virus replicates some more. After that, there's a viremia. The virus spreads to other organs, including spleen. And we know that sensing of the virus by TLR3 leads to the production of TNF-alpha. That loosens up the blood-brain barrier, lets the virus get in the brain and cause that neurological problem in a small percentage of individuals. Interferons appear to be important for regulating infection, and neurons in the CNS are the main target. This slide has a lot of pictures of T cells and chemokines that are important in regulation of the infection. The key point here is that the virus can get into the brain because of the innate immune response. And the rest of the West Nile fever is caused by the reproduction outside of the CNS and the induction of cytokines and chemokines that give you that West Nile fever, if you will. Here's a graph and table of West Nile in the U.S. from 1999 when it first came to the U.S. To 2019. We're looking at the number of cases and which are neuroinvasive in the light purple and non-neuroinvasive, and deaths in orange. And you can see it varies according to year. Some year very low, some year very high incidence. One hypothesis is that on very dry years, the birds go away from wherever you are, and the mosquitoes don't have birds to bite, so they bite people instead, but I'm not sure that's correct. And on the right here is the seasonality of West Nile in the U.S. Number of cases with the week of onset. You can see starting in June and ending in November. This is the time when the mosquitoes are out and biting, so it corresponds precisely with the time mosquitoes are most abundant. And this is a summary of the annual incidence of the neuroinvasive disease by U.S. county from 1999 to 2018, and the darker colors are higher incidence. And this simply tracks with the number of infections. Areas of the U.S. with the most mosquitoes tend to have the most infections and the most neuroinvasive disease. We have relatively very little here in the Northeast. And finally, the neuroinvasive disease by age group. And you can see this is increasing as age increases. And so the ability to invade the CNS is clearly associated with a decline in immune functions, but there's not zero neuroinvasive disease at younger ages. What can you do? Until we have a vaccine, you should just avoid mosquitoes. This species bites from the evening to the morning. So if you're out and about that time, you should use repellents. Make sure there's screens on your homes. Wear the proper clothing in those months. Obviously, the other months is very much less risk, and we should have vaccines available. This is why we have a horse vaccine. It has a 30% fatality rate in horses. And of course, they're vulnerable sitting in their stalls to being bitten by mosquitoes. I want to end with just a few points about viruses in the central nervous system. I've talked about 3 viruses today, poliovirus, measles virus, and West Nile virus, that invade the central nervous system. But that, in my view, is a dead end because these viruses are all really well transmitted by shedding elsewhere. Poliovirus from the gut, measles from the respiratory tract, and of course mosquitoes are transmitted. West Nile virus is transmitted by mosquitoes. So the CNS is a dead end. When a virus gets in there, it doesn't get out again. And so not only is it a dead end, but there can be no selective pressure for a virus to evolve to go into the central nervous system. Often when a virus emerges with a property of causing CNS disease, and this has happened for a number of viruses. For example, Zika virus was known since 1947, but not till 2015 did we see CNS infections. And people said, oh, it mutated to enter the CNS, but that can't be because there's no selective force for entering the CNS. There's only selection for transmission to another host. All right, so that's acute infections. Next time we're going to talk about viruses that cause lifetime infections of the host, which is persistent infections. And let's see if we have any more questions. Sorry. The disease has 30% fatality in horses. If the vaccine had 30% fatality, that would be very bad, I think. I should fix the slide. If older people have weaker immune systems, wouldn't they produce less TNF alpha, so less permeable? Why would older people have? Because probably they have higher levels of virus, and even though they're making a less of an innate response, they have still more of a problem because there's a lot more virus in them. With respect to CNS being a dead end, how can rabies be explained? Is the CNS infection just a random thing that was not selected for? Well, it's the same as with polio or West Nile. The virus accidentally gets in the CNS. I think it is. It's not a human. Rabies is not a human virus, right? It's a bat virus, essentially. And it's in the wrong host, and it just finds itself entering the CNS, probably because there are receptors and the cells are permissive. So even if there is no selection for CNS, couldn't there be a mutation that it happens to increase? Yes. So it could be some other mutation that's done something else to, say, increase respiratory titers or something like that to get better transmission. And that accidentally causes CNS invasion. Yes, that's fine. But to say that CNS invasion was selected for is not correct. That's my main point. Wouldn't rabies selectively invade the brain to change their behavior to increase transmission? Not in people. We typically don't transmit it to each other. You get pretty debilitated by rabies, and you end up hospitalized, and eventually it's almost 100% fatal. So in animals, yes, it could be that, yes, animals who eat each other, for example, it could have been selected for. So my issue with the CNS as a selection for invasion is just in people, right?